Go ahead, David.
Ready to begin? Okay.
Yeah.
Good evening. I'm David Feldman. I'm one of the founding family members of the Children's Fund for Glycogen Storage Disease Research, an organization that we started back in 2002, and we continue still to fund the world's best GSD research still to date. So we'd like to welcome you to the GSD webinar, hosted in partnership with our foundation, the AGSD and Ultragenyx. And the purpose of this forum is for the GSD community to learn about research underway at Ultragenyx.
Thank you, David. I'm Iris Ferrecchia. I'm the president of the Association for Glycogen Storage Disease. We actually collaborate with family organizations all over the world, and so I know that I see our numbers are growing. We have over 121 on this town hall meeting right now, and I am really grateful for the interest. And I just wanted to say one thing, that even though we are hosting this town hall, we're not endorsing any specific program or suggesting any participation in these projects. This town hall is for informational purposes only. And I'm grateful for your interest because I'm sure after this we'll have a lot of discussion on Facebook. And I just wanted to say that the event is being recorded. So for those friends overseas who maybe couldn't stay up so late, they may be able to visit it in a different way later. I'd like to also just thank Ultragenyx for taking this time to make this available to families and patients who live with glycogen storage disease 1A and 3 alpha. Thank you.
Thank you, Iris. Thank you, David. My name is Andrew Grimm. I'm a pediatric gastroenterologist, physician scientist trained in genetics, and pediatrics, and gastroenterology, and I've been doing rare disease drug development for my whole career so far. I am honored to have the opportunity to speak to the GSD community, and especially thankful for the support from the Children's Fund and AGSD in setting up this town hall. We are striving to make sure that the GSD community is aware of the efforts that we're putting in to develop new therapies, and we're happy to share with you where we are right now. And as you'll see during the webinar, there's a lot of uncertainty still in the future, and what the studies will show, that we're excited to give you an update. So with that, if we can go to the next slide. I work for Ultragenyx, as does Dr. Vagilanopoulos, and we both joined the company pretty recently. And one of the things that drew me to Ultragenyx was the focus on rare, severe, unmet needs in genetic disorders that were addressable with therapies that are now being developed very rapidly. The company was founded in 2010, about 10 years ago, and it's been growing rapidly under the leadership of Emil Kakis, who founded the company. We're focusing on diseases where we have a clear understanding of the biology and where there's no approved therapies. And it's a really exciting time to be working on these kinds of projects where the technology is advancing so rapidly. And as you'll see in some of the discussions today, the advancements in science are now making it possible to treat diseases that we previously really didn't understand the molecular basis for just a few years ago. Next slide. So the mission of the company is to develop treatments and to make them as widely available as possible for rare and ultra-rare diseases. And it's relatively unique in the sense that the company takes a relatively agnostic approach to how we're going to move the field forward, and are willing to partner and with academic institutions and with foundations. And the highest priority is getting new therapies developed for patients and finding creative ways to do that. Next slide. So with that, I'm very pleased to introduce Dr. Valianopoulos, who's an expert in glycogen storage diseases, who will give you some background on the GSDIa program.
Thank you very much, Andrew. And also I'd like to thank our partners tonight for the invitation, the opportunity to speak about GSDs in general, but more specifically GSD1 gene therapy programs. A few words about myself. So I'm a metabolic pediatrician by training. I worked more than 12 years in clinical practice at Necker University Hospital in Paris, caring for patients with various metabolic diseases. So I've seen and treated patients myself with GSD1a and GSD3. So it's a new excitement for me to be able now to be heading up this program from the clinical development standpoint and to build all the clinical program that hopefully will make this new approach, this new gene therapy approach, a valid treatment for GSD1a. And before we get more into the details of gene therapy, I just wanted to mention that tonight we're going to be talking to you about two different glycogen storage disorder types, so GSD1a obviously and GSD3. And besides the fact that these are both genetic diseases that involve glycogen breakdown in the liver and various tissues, they have also significant differences. And that accounts also for the differences in terms of approach and therapeutic modalities, but also for the potential endpoints that we use in our clinical trials. So in other words, it's expected to see differences between the two programs. And even though the root cause of the problem, which is the genetic defect, is going to be addressed directly or indirectly, the ways to do that might differ. And I wanted to be upfront with you so that you understand the similarities, but also the differences between the two programs. And with that, I would propose to move to our next slide, which is a kind of introductory slide on the principles of gene therapy. So I put together this simple cartoon that illustrates these principles of gene therapy. So it involves different steps, as you can see it here. So the first step is choosing a gene of interest, which is generally the one responsible for the disease. The second step is to insert the DNA sequence in a gene therapy vector, which is generally a virus. For instance, a lot of these therapies, including the one we're developing for GSD1a, is based on the adeno-associated virus, or for short, AAV, where the virus's own genetic material has been removed. And then once this viral vector is loaded with the gene of interest, DNA, then there is phase 3, which is an administration of this viral vector, generally through injection, which can be local or systemic. In this case, it's generally an intravenous injection. And then what happens is that this AAV is infecting the cells, and in a way, this AAV will be targeted to the tissue of interest. So if liver is our interest, then we'll choose a type of AAV that principally meets liver cells. If it's the muscle and the liver, then we might use something different, and so on. But once this AAV hits its target and infects the cells, it will release its genetic material, because this is what viruses do. That will go into the cell nucleus and form what we call an episome. So it's a sort of circular DNA that can be used as a template to produce the deficient protein that will be used by the cell, or can be secreted and uptaken by other cells. And depending, as I was saying, on the type of vector used, different types of cells or organs can be targeted by using specific capsids that have a preference for one or other cellular type. Some viral vectors, and this is the case mostly of the lentivirus, can be used for ex vivo gene therapy, where patient own cells, where generally precursor of blood cells, are extracted, transduced, or manipulated to transfer the gene of interest and then re-injected in the patient's circulation. This is not what we're going to be talking about tonight. We're going to talk about AAV-targeted gene therapy, in which the construction and the delivery is much simpler than the lentiviral gene therapies. So if we go to the next slide, just to present what our investigational product looks like. So this is called DTX401. So it's a gene therapy product that was designed specifically for GSD1A. So what is it? It is a non-replicated recombinant AAV of a specific serotype, which is a serotype 8, that allows this vector to target mainly liver cells and not go elsewhere. And so this product is encoding the human glucose 6 phosphatase catalytic subunit, which is the deficient gene, as you all know, in GSD1A. So based on animal studies and other toxicology studies and multiple experiments, DTX401 has been shown to be able to deliver the gene of interest to liver cells, hepatocytes, resulting in an improved glucose 6 phosphatase activity. And that's how it is created as a potential drug to correct the underlying cause, which is a genetic defect in GSD1A. So these are the principles of gene therapy. And obviously, I'm not going to spend more time discussing about the specifics of the disease that we're trying to address. But I want to dedicate this time in your questions, and I will invite David to ask the first question, and then we can go through the list.
Thank you, Dr. Vig. Why did Ultragenyx choose GSD1A for your first gene therapy program?
That's a very good question. And I think the general way to approach this question, and that applies to most of the diseases, there are a lot of factors that influence this decision. One of the key ones is how much unmet need in a specific disease there is. And for instance, we have many other programs that address diseases in which there are no therapeutic options currently, or very few. So that's one key factor. Then it's also the ability, the technical ability, the size of the gene to be delivered in an AAV vector when it comes to the modality of the gene therapy. It's also the fact that the key feature of this disease is hypoglycemia that can be readily measured and used as an endpoint to show efficacy in a clinical trial. So in a way, hypoglycemia becomes the surrogate of drug efficacy. But I would say one of the most critical factors is the demonstration that this is a safe product through extensive testing in animal models, and that it can be safely delivered in a patient population in need. So I think these are the key factors that led to selection of GSD1A as a candidate disease for a gene therapy program.
How is this therapy administered, and how many doses are needed?
That's a very good question. So as I just mentioned in the general principle for AAV liver-targeted gene therapies, the administration is done through a single intravenous infusion. That means the patient receives just one dose of the therapy, typically through a vein in the arm. And once a patient is exposed to the AAV serotype used for gene therapy, then the patient generally develops antibodies to the serotype and can no longer receive gene therapy utilizing that serotype.
At the end of phase 3, how long will it take for patients to have access to the therapy?
This is another very good question. So I'll start with a general statement that access to therapy outside the context of a clinical study is only possible after regulatory agencies' approvals in their respective countries. So Ultragenyx is continuing to collect longer-term data from the ongoing phase 1/2 study, which are expected to be available towards the end of the year, bearing further significant delays related to COVID-19. So that's rather good news. Also, We recently held an end of phase 2 meeting with the US FDA and a meeting with a protocol scientific advice working party in Europe. And the phase 3 study, based on the interactions with these agencies, is still expected to begin in the first half of 2021, depending on the outcomes of the discussions with the agencies and whether there are further COVID-19 related delays.
Have you decided where the clinical trial sites will be located for phase 3?
We're actively working on this part. Obviously, since I just announced that we're heading towards the beginning of our phase 3 in the first half of 2021, we start preparing the sites where we plan to go. So the fact is that we're planning to go more international and more global than our current phase 1/2, and choosing more countries and more centers within a country. And obviously, to be included in a study, center must fulfill a series of criteria, especially related to the structure that includes the procedure for carrying out gene therapy and also being able to adequately monitoring patients.
How are the patients who receive gene therapy doing currently?
I will start with a general statement that we make in these, because these are confidential data and that on an ongoing trial, we can only disclose what has been being publicly available. And even though this might sound a bit old already, the most recent results we have from a phase 1/2 study communicated were from May 2020 at the American Society of Gene and Cell Therapy. So these data that were presented by Dr. David Weinstein demonstrated that all patients at that time point had improved response in their cornstarch requirements, and most patients in their times to hypoglycemia, and that DTX401 had an acceptable safety profile across all patients treated in all dose cohorts.
If the therapy is successful and approved, would we expect an improvement in GSD or a cure?
I think this is a very important question, and I'd like to remind that since GSD1A is an inherited genetic disease, every cell in an affected individual's body contains the mutations. Gene therapy is designed to correct the mutations in part of the liver cells, enough to restore part of the liver's affected function, and therefore may help control disease clinically. As the other cells still will harbor the mutation, the word cure would not be appropriate in this context.
Is the therapy being tested only in adults, or is it also being tested in children?
In phase 1/2 study, only patients 18 years or older were enrolled. Thus, until now, this potential gene therapy has been studied only in adult patients. For phase 3, the possibility to include patients with GSD1A under the age of 18 is being considered, but will need to be discussed with the health authorities.
Can patients with GSD1A who have received the therapy get it again? If not, why?
Yes, this is another important question. So patients who have already received gene therapy with DTX401 are likely to have developed antibodies against the vector that would significantly prevent efficacy from happening and would not be able to receive a second dose for now. It is not excluded in the future that Ultragenyx, as other companies in the gene therapy field, might find different solutions for that problem. But as for now, there's only one dose that will be administered in patients in clinical trials.
What are the side effects related to the gene therapy?
So far, again, based on the data that have been publicly disclosed, there have been no infusion-related adverse events, no treatment-related serious adverse events throughout the phase 1/2 study. 7 out of the 9 patients included in the study had mild asymptomatic elevations of enzymes related to liver function. These were mild and transient for most patients and similar to what has been observed in other AAV gene therapy programs, and they have been successfully treated in all patients with steroids in a reactive manner. Additional safety information will be collected in the planned phase 3 study, and obviously the safety of the product will be fully established.
What is the proposed mechanism of action of DTX401?
I think it's exactly as I illustrated in my example. So it's an AAV8 gene therapy product designed to deliver stable expression and activity of the glucose 6-phosphatase enzyme under the control of its native promoter. So it is administered, as we mentioned, as a single intravenous infusion, and has been shown in early studies to improve glucose 6-phosphatase activity and reduce liver glycogen levels, which is a well-described biomarker of GSD.
What is the estimated cost of the gene therapy for GSD1a?
Yeah, the cost model for gene therapy has been discussed worldwide and by many companies. Gene therapy development costs are relatively higher than traditional therapy, and the dose is usually single. And so this makes it more challenging to predict the reimbursement model than in other therapies. And as this therapy is still in development, it's difficult to estimate any market cost for it at this stage.
Dr. V, will you be using a placebo in this study? If so, why?
Yeah, this is another important question when it comes in terms of clinical trials in rare diseases. So I'd like to mention that the design has not been finalized, and a lot of these discussions are ongoing with the regulatory agencies. So the FDA has explicitly requested for a placebo-controlled study as a gold standard for phase 3 clinical trials. And if the phase 3 is a placebo-controlled study as requested and as our initial plans are, we're planning to offer gene therapy at a crossover point later in the study for those patients initially randomized to placebo. So exiting this study, all patients will have been treated with a gene therapy product. Different timeframes, but they will all be treated eventually.
Great, thank you. What kind of support is given after receiving the gene therapy, and for how long?
It is obviously very important that all patients treated with gene therapy are followed throughout time to establish the long-term efficacy and especially safety for the product. So all patients participating in a clinical trial will be asked to participate in a disease monitoring program, which is an observational study where they will be seen regularly at the regular pace of follow-up with their treating physicians. And most of the assessments will be tailored to their standard of care.
What will be evaluated in phase 3?
So these are things that are under consideration and discussions with the agencies, but we can reveal some of the things we're thinking about. So cornstarch requirements that we've seen in phase 1/2 responding very well is one of the key targets here. Obviously, we want to see this happening without having a negative impact on glycemic control, and even improving glycemic control is likely to be one other of our endpoints. We're likely also to repeat the fasting challenge test because they're very good indicators on fasting tolerance that tends to increase over time after treatment. And some of the things that we'll implement in this phase 3 is the way we collect data. So continuous glucose monitoring that has been implemented in phase 2 will continue in phase 3, and some specific electronic devices that will allow to capture cornstarch and diet in a systematic way are some of the plans for our phase 3.
How many people are you accepting into the phase 3 trial?
Yeah, this is always a challenging question, and we always aim to have the minimum of patients that we need for this type of investigation. So we're looking into a 40-patient study right now, but it's not excluded based on our conversations with the agencies that we might go higher or lower, depending on what the final selection criteria will be for these patients.
What are the inclusion, exclusion criteria for phase 3?
Yeah, as I was mentioning earlier, some of these things are under discussion with the agencies. So the plan for right now is not going to be very different from the inclusion-exclusion criteria in our phase 1/2 trials. And I invite everyone to go on clinicaltrials.gov and read those in detail. But what I wanted to mention that there's one key exclusion criterion that we will have to follow, which is the presence of neutralizing antibodies against AAV8, because as I explained earlier, it prevents efficacy from happening. So that's probably one of the most certain criteria. And when we'll have final agreement on the study design, phase 3 key inclusion-exclusion criteria will be posted on clinicaltrials.gov, and they will be publicly accessible for everyone.
How long is the gene therapy expected to last?
This is a question that the regulators ask us all the time and anticipate they will continue asking. So far the response we're providing is that it's obviously too early to address the question of the durability, but it has been more than 2 years now since the first subjects were administered a single dose of DTX401, and their results have remained consistent over time.
Who has taken over for Dr. Weinstein as lead investigator?
Yes, since early August, Dr. Rebecca Riba-Wollman, who's a pediatric endocrinologist, has taken over the responsibilities of Dr. Weinstein at UConn.
Will GSDIa patients be able to drink alcohol, take Tylenol, or use other products that can be harder on the liver after gene therapy?
Yeah, I think I understand the anticipation and the question, but it's probably too early to answer this question. And what I would say is that based on our preliminary safety data, gene therapy does not make liver more vulnerable, but we don't think it will make it more resistant as it is. So patients should not anticipate any different effects of toxic substances in the liver before or after gene therapy.
Will exercise or extreme temperature environments still affect GSDIa patients via high lactates and hypoglycemia after gene therapy?
Another great question, another answer that will be similar, that's probably too early to be able to conclude on this type of question. We have seen several patients in the phase 1/2 study who have reported increased ability to exercise without experiencing adverse events. However, we think that safety-related measures, including avoidance of extreme situations, should remain the rule even when gene therapy is approved and administered, and until we have sufficient data on the level of metabolic correction that can occur sooner or later in different subjects.
Can I pass GSDIa to my offspring after gene therapy?
Yes, let's make it clear here, the risk will remain the same. Liver-targeted AAV gene therapy do not impact germline cells. And this is obviously a safety requirement for most gene therapies, not to alter germline cells, because obviously they could lead also to inherited traits in the offspring. So no change after gene therapy on the risk to have an offspring with GSD1A.
Dr. V, thank you very much for those answers.
You're very welcome. Pleasure. Andrew, are you saying something? We can't hear you very well. I think we might have a small technical sound issue here.
Hello?
Oh, there you go. Now we can hear you.
Okay, great. Thank you, Vasily, for going through the GSD1A program. If we could go to the next slide. Forward one more. Okay, so I'm going to give some background about our program in Glycogen Storage Disease type 3. And we're using a different approach. It's similar, but it has some important differences that I'd like to highlight. So next slide, please. So there's been a lot of excitement recently about mRNA-based therapies. You may have heard about them in the setting of development of COVID vaccines. This has been really nice for us to increase awareness about mRNA-based therapies. And the basic idea is that mRNA is how the body tells the ribosomes in the cells to make proteins. So the mRNA is a messenger, right? It's a unstable intermediate between the gene that encodes the protein and the functional protein itself. And in the case of GSD3, the mRNA that we're trying to deliver is the one that encodes the glycogen debranching enzyme on the AGL gene. But you could deliver an mRNA for any number of genes. These mRNA intermediates are, as I mentioned, unstable, and they need to be protected. And we need to get them into the tissues so they can produce the missing proteins. So we encapsulate them into a lipid nanoparticle. So this has several different types of lipids, types of fats on the outside that help to stabilize it and give it a structure, and also help it get taken up into tissues, and then to deliver the mRNA into the cells. These infusions, this is given by an IV infusion. You can't, or in the case of vaccines, it's given intramuscularly. But in the case of our approach, we're using an IV infusion so we can deliver it to the whole body. Next slide, please. So after it's given IV, it goes into the blood and then it goes into cells. And the lipids on the outside bind to receptors on the cell membrane. And then this nanoparticle gets taken up into the cells, into an endosome. And then the endosome pH changes, it drops. And that helps to activate some of the lipids on the lipid nanoparticle that release the mRNA from the endosomes. The endosome breaks open, the mRNA is released inside of the cell. And then the ribosomes, which are the machines that translate the mRNA into protein, get to work and they produce the protein. So in this diagram, the protein looks like a little purple Pac-Man. But the idea is that the protein then does the job that it's supposed to do. And in the case of glycogen storage disease type 3, it's the debranching enzyme that then helps to complete the breakdown of glycogen by resolving the branch structure. Next slide, please. One important difference between gene therapy and an mRNA-based therapy is that the mRNA is just an intermediate. It doesn't stick around for very long, and so it has to be given on a regular basis. Gene therapy is designed to stay in the body, and the DNA gets copied, gets transcribed into mRNA over and over again. So over many months to many years, more and more mRNA gets produced, and then more protein gets produced. In the case of an mRNA therapy, because we're not giving the underlying gene, the mRNA has to be given on a regular basis. So one of the benefits of an mRNA-based therapy over a gene therapy is that you can adjust the dose as needed. You can start with a low dose and then go up on the dose and try to find the right dose to minimize side effects and hit the right efficacy sweet spot. Obviously, the repeated dosing can be inconvenient, and giving regular IV infusions is more burdensome than giving a single IV infusion. So there's benefits and drawbacks. We're not using a viral vector, so issues of antibodies, immune responses to the lipid nanoparticle don't apply initially. Still, you have the potential in any case to develop an immune reaction. Obviously, in the case of COVID vaccines, you're trying to stimulate the immune response to develop antibodies against the viral proteins. That's not what we're doing in this case. I think this is the last slide that describes the strategy that we're taking to try to replace the missing enzyme in GSD3. And Iris is going to be relaying some questions that we got from the community, and I'll be filling those questions.
Thank you, Andrew. First question is, why did Ultragenyx choose mRNA for GSD3 instead of gene therapy?
So the glycogen debranching enzyme is a very large protein. It has two different catalytic activities. There's a glucosidase and a glycosyl transferase activity in the same protein. And the gene itself is quite large, and it's too large to fit into a regular AAV-based vector. So the primary reason that we're taking the mRNA-based approach is because you can put much longer mRNAs into a lipid nanoparticle, and you can produce a larger protein. And as I mentioned, there's other potential benefits about adjusting the dose and potentially in having a wider tissue distribution, depending on which cells are taking up the lipid nanoparticle.
Thank you. What is the proposed mechanism of action of the UX053?
So I hope that was clear from the slides that I presented. The idea is that the mRNA that we're delivering is translated into the functional glycogen debranching enzyme protein, and that's replacing the missing protein in patients with GSD3, and that will allow glycogen to be broken down normally in the cells where lipid nanoparticle penetrates.
Okay, thank you. How is this therapy administered, and how many doses are needed?
It's given intravenously, and we don't know exactly how long the enzyme is going to stick around in humans. We're doing animal testing. It may be once every 2 weeks, maybe once a month. It's hard to predict at this point because we haven't started our clinical trials yet. We'll be collecting data on the blood levels of the mRNA to track how often the dosing should be given.
When will Ultragenyx begin recruiting patients for the GSD3 clinical trial?
So we're planning to file our investigative new drug application in early 2021. And if cleared, then we'll initiate our first human study in early next year.
Do you know where the clinical trial sites will be located?
We're looking worldwide at a number of clinical trial sites. We haven't finalized the site selection process yet, so I can't tell you all the locations yet.
What are the side effects related to the mRNA therapy?
So since we haven't tested our therapy yet in patients, we don't know for sure. We can say from similar molecules where lipid nanoparticles are given intravenously, infusion reactions tend to be the most common side effect, and those could be activation of the immune system that could lead to flushing, nausea, vomiting, body aches, headache, rash, chills, or dizziness. We don't know which of those side effects we might observe in our studies.
Is the therapy being tested only in adults, or is it also being tested in children?
So our first study, for ethical reasons, will just be in adults, but we recognize that there is a substantial burden of disease in children and a high unmet need. And as a pediatric gastroenterologist, I'm very motivated to get a therapy approved for children. So we will, if the first in human study in adults is successful, we would then proceed to pediatric development too.
If the therapy is successful and approved, what benefits would we expect from treatment?
Well, we don't know for sure if we're going to get how profound the effect will be because we haven't tested it yet in patients. Our hope is that it will reduce glycogen storage dramatically, especially in the liver, and will allow patients to reduce their cornstarch intake. Hopefully children who have slow growth will have improved growth. It hopefully will help provide energy supplies for muscle. We know that the muscle disease can be quite substantial on GSD3, but whether we can affect all these processes remains to be seen because we haven't tested it yet.
Do we know when mRNA therapy will be available for patients with GSD type 3?
It's hard to predict. It's drug development is a long road. I think looking at the COVID trials, it's a very unusual situation. We're not going to be able to get a drug, to market within a year. It's a long process, but we are committed to getting it done as quickly as possible, and really depends on how quickly we can recruit patients in the study, how quickly we can analyze the data, and also depends on regulatory interactions. And so there's a lot of unknowns, and it's too hard to predict at this point if this drug would get approved.
Thank you.
One thing I'd like to add, tangential to the discussion we had, is Ultragenyx is working with a company called Glycogen Health to help understand the journeys that patients with glycogen storage disease have. And there's a program called the GSD Odyssey, where patients can have all their medical records collated and all at one site, so they can help navigate the healthcare system and understand all the different specialists that are involved in managing their care. There is a link to this program on the Ultragenyx Facebook page. So there's a post there. If you find the Ultragenyx Facebook page, you'll see a link to the GSD Odyssey program and some more information about that. It's a tool both for patients and a research tool for scientists who are trying to understand how patients with GSD3 navigate the healthcare system and how the disease is affecting them. So if those are all the questions, I want to again thank David and Iris for allowing us to talk about the programs that we're developing at Ultragenyx. And I'm really excited to have the opportunity to share where we are with the GSD community. And it is a really exciting time to be involved in these programs and seeing the data move forward and seeing potential new therapies in the horizon.
Thank you, Andrew. And I hope I'm not jumping the gun. I just wanted to thank all of you for the work that you're doing and for making yourself available for this kind of a interchange. And I wanted to just ask the people in attendance to, if they wouldn't mind, or if they have access to my contact information, just give me their feedback if they are interested in more types of interactions like this, especially during COVID, when we could meet virtually in this kind of a venue. Thank you again. David?
Yeah, I want to thank Kristen, especially Kristen Smith from Ultragenyx, for listening to our idea and making this event happen. I want to thank Cyril and Mindy behind the scenes, Vasily, Andrew. As a GSD parent, we are very lucky to have you guys lead the charge of these studies. So thank you.
It's a pleasure and an honor.
Same here, and we're all excited to have this very important partnership with you all.